Have you ever heard about the Smart Plant-Wearable Biosensor for In-Situ Pesticide Analysis? We from Group 8 will present about this particular topic, and these are the group members. The issue starts when in recent years there's a significance of advancement have been made in stretchable and flexible electronics. So among these advancements, in wearable biosensor, it actually have gained considerable attention because of their ability to provide continuous, non-invasive and real-time monitoring during dynamic analysis. However, the issue starts when the majority of the wearable biosensor have predominantly applied in fitness and healthcare industry for the monitoring of the tracking ion and molecule, for example. So, recognizing the global demand for food sustainability, researchers have begun focusing on plant sensors to enable sustainable agricultural production. Plant sensors are promising devices in agriculture as they can collect real-time information about the plant physiological status as well as mechanical compatibility to allow them to easily attach to soft plant surfaces. These sensors have been explored for monitoring plant growth, relative humidity, plant disease, and providing crucial real-time information about plant health and specific needs. Additionally, the assessment of sensing agrochemical, such as pesticide, for example, is essential for the advancement of the precision agriculture. And despite this development of various analytical technology for pesticide detection, many still require some complicated sample treatment, which does not meet the need for rapid and non-destructive analysis. So in order to address this challenge, in situ detection methods such as surface-enhanced Raman scattering on crop surfaces have been developed. However, there has been a limited research on electrochemical technology for in situ pesticide analysis on crop surfaces.
Hence, the concept is to develop a smart plant wearable electrochemical biosensor for in situ analysis of organophosphorus pesticide, specifically methyl parathion on crop surfaces. By capturing and recognizing pesticide molecules, the biosensor generates an electrochemical signal proportional to the concentration of the pesticide present. The biosensor element is an enzyme that selectively captures and recognizes the target pesticide, namely organophosphorus hydrolase. NAF-ion is a biocompatible polymer widely used as an electrode modifier that promotes the immobilization of the organophosphorus hydrolase enzyme on the electrode surface. NAF-ion also helps in stabilizing the enzyme and preventing interference from other substances. For the transaction method, the laser-induced graphene-based electrode acts as the transducer by converting the biochemical reaction into an electrochemical signal, which is then measured using a handheld electrochemical workstation. The target molecule that can be detected using Smart Plant Wearable Biosensor is organophosphorus pesticide, specifically methyl parathion. Therefore, when the target pesticide methyl parathion is present, the immobilized organophosphorus hydrolase on the electrode surface catalyzes its hydrolysis to produce p-nitrophenol, which is electroactive and can be detected by the biosensor. This is the schematic diagram of the working principle of the Smart Plant Wearable Biosensor. Firstly, the biosensor is attached to the crop surface such as spinach or apple using its flexible PDMS backing. Then, a layer of gelatin semi-solid electrolyte is applied between the sensor and the crop surface to ensure proper contact and facilitate the electrochemical reaction. When methyl parathion is present on the crop surface, it diffuses into the semi-solid electrolyte and comes into contact with the organophosphorus hydrolase enzyme immobilized on the electrode. Organophosphorus hydrolase hydrolyzes methyl parathion to produce p-nitrophenol. The reaction is facilitated by the presence of gelatin electrolyte, which provides a suitable medium for enzyme activity. The biosensor sensitivity is enhanced by the 3D porous structure of laser-induced graphene and presence of gold nanoparticle on electrode, which increases the electrode surface area and conductivity. The biosensor selectivity is achieved through the specific enzymatic reaction of organophosphorus hydrolase with methyl parathion, which means organophosphorus hydrolase will only hydrolyze methyl parathion to produce p-nitrophenol. The produced p-nitrophenol is then oxidized or reduced at the electrode surface, generating an electrochemical signal proportional to the pesticide concentration. This signal is measured using square wave voltammetry as the electrochemical measurement technique, with specific parameters include frequency of 8 Hz and equilibration time of 30 seconds. The electrochemical data is transmitted to a smartphone using a handheld chemical workstation with Bluetooth, where it is processed and displayed, allowing for real-time monitoring and analysis of pesticide residue. This analytical strategy eliminates complicated sample pre-treatment steps and enable rapid, non-destructive in-situ pesticide recognition on crop surfaces. The detection limit of the biosensor for in-situ analysis of methyl parathion is 0.01 micromolar, meeting the regulatory demands for methyl parathion residue levels in vegetable and fruit set by the European Commission.
The biosensor can serve as an early warning system by monitoring pesticide levels in crops. The study of plant-wearable biosensor could certainly have application in quality control, especially in industry related to agriculture or environmental monitoring. For the application in environmental monitoring, by detection high intensity of chemical compound in certain area, we can control the indiscriminate use of pesticides. Farmers can target specific areas or crops that require treatment, minimizing chemical exposure in the environment and reducing the risk of pollution which is harmful to plants and animals. For examples, soil, air, and water pollutions. In the application for agriculture, this biosensor enables farmers to quickly and non-destructively assess the presence of harmful pesticide on crops for ensuring compliance with regulation to avoid any food poisoning to customers. For example, phosphorus, chloride, and nitrate which can cause baby blue to birth. But how this biosensor can detect the presence of certain compound of these pesticides? The biosensor provides farmers early sign of plant disease through various indicators such as changes in electrochemical signals or the presence of specific biomarkers. For example, the higher use of phosphorus and over-fertilization, which can detect by the electrochemical signals that can cause leaf tick-borne disease to plant.
So there are first alternative solutions to enhance biosensor performance so that it can be applicable in the agriculture industry. The first one is developing multiplex capabilities. The second one is improving scalability and reproducibility. The last but not least is integration with data management systems. So for the first one, which is developing multiplex capabilities, the current biosensor is designed to detect only a single type of pesticide, specifically methyl parathion. This limitation restricts its utility in diverse agricultural settings where multiple pesticides and other parameters also might need monitoring. To address the limitation of single anomaly detection, developing multiplex sensor can be beneficial. This sensor will be capable of simultaneously detecting multiple pesticides and other relevant parameters, such as nutrient level and plant stress marker. This can be achieved by incorporating different recognition elements and transduction mechanism within a single sensor platform. So the second one, which is to improve scalability and reproducibility, the fabrication process of biosensor, which involve laser-induced graphene or LIG and polydimethylsiloxane or PDMS, face challenges in scalability and reproducibility. Ensuring consistent quality and performance across large-scale production is currently very difficult. To overcome the challenge in scalability and reproducibility, developing standardized and automated manufacturing process is very essential. This including optimizing the LIG fabrication by adjusting laser power, scan speed, and other parameters to produce consistent porous LIG structure with high conductivity. Additionally, establishing uniform methods for transferring LIG to PDMS is very beneficial. Integrating biosensor with advanced data management system can further enhance their utility. Implementing machine learning algorithm will allow for the detection of pattern in the data and prediction of the issues such as disease and nutrient deficiencies, which provide valuable insight for the agriculture manager.
In conclusion, this electrode system easily patents using laser-induced graphene LIG technology by controlling laser power and scan speed. After being transferred with PDMS, stretchables and flexible LIG-based electrode can be prepared. This can maintain the satisfactory electrochemical response during process of stretching and bending. The smart plant wearable biosensor modified with OPH and equipped with gelatin electrolyte can be attached to the face of agricultural products such as spinach leaf and apples. This allowing real-time or on-site analysis of methyl parathion by chemical detections of hydrolysis product. This will give us new insights, designs of next generation wearable biosensor for future agriculture.